So here we're going to overview glycogen phosphorylase regulation. But before we jump into that, we need to recap, what does glycogen phosphorylase do? We have to remember that this is an enzyme, and the function of this enzyme is to cleave terminal glucose molecules within glycogen to produce glucose 1-phosphate, which can be converted into glucose 6-phosphate that can be used by glycolysis for energy when we have low glucose levels in the body. So once again, glycogen phosphorylase is used to break down glycogen. Specifically, it's going to break the terminal glucose molecules, attach a phosphate to it. It's going to become glucose 1-phosphate, and then we'll see that conversion into glucose 6-phosphate and then into glycolysis. But with any enzyme in the human body, of course, we need regulation. And here we're going to go over how this enzyme is regulated. So if we take a look at this enzyme, it exists in two different states. It can exist in an inactive or a relatively less active form, or it can exist in an active form. Now, the active form is going to be phosphorylated, whereas the inactive form is going to be dephosphorylated. So within glycogen phosphorylase, remember, it is an enzyme. Enzymes are proteins. Proteins are made up of amino acids. Specifically, within our glycogen phosphorylase, we have a serine residue. And on the serine residue, that is where the phosphorylation is going to occur. How do we phosphorylate? Well, we have to utilize a kinase enzyme. When we utilize a kinase enzyme, what do we need? We need ATP. Specifically here, we need 2 ATP molecules. So we take 2 ATP molecules, we phosphorylate 2 serine groups onto our glycogen phosphorylase. As a result, we produce 2 ADP. Because ATP is triphosphate, now we have diphosphate because we've taken a phosphate off of each ATP. Now we have phosphorylated glycogen phosphorylase, and we are in the more active form. Now, when we take a look at textbooks often refer to this phosphorylated form as the R state, also known as the relaxed state. So R stands for relaxed, and this state refers to the phosphorylated, active, relaxed state of the enzyme, whereas the T state is the tense state that is unphosphorylated, and it is inactive or relatively less active when we compare it to our more active form. So going back to our kinase, the kinase, when is it activated? The kinase will be activated when we have glucagon present or we have epinephrine present. What does this mean? Glucagon means we're going to have low energy levels, and essentially we have low glucose levels. So if we have low glucose levels in the body, or we have low ATP levels in the body, and we have higher AMP levels in the body, we need a source of energy. And remember, the first form of energy is, of course, glucose. The second form of energy is fats. And the third form of energy utilized by the body is protein. Where do we store glucose for energy? Well, we store it as glycogen. And now when we have low glucose levels, we're going to release glucagon. And as a result, we're going to phosphorylate the glycogen phosphorylase. When we phosphorylate it, it becomes more active, and it's more readily going to break down glycogen to get our glucose 1-phosphate. So essentially, the kinase is activated by glucagon and epinephrine. How does epinephrine take a role into this? Well, remember, epinephrine is produced in our fight or flight response, and in that case, we are going to need more energy. And when we need more energy, we're going to need more glucose. Now, glucose acts as an inhibitor of glycogen phosphorylase, and this makes sense because, once again, why do we need glycogen phosphorylase? We need glycogen phosphorylase to break down glycogen into glucose 1-phosphate monomers. But let's say we have more than enough glucose in the body. We have lots of glucose. What are we going to produce? We're going to produce insulin. When we produce insulin, because we have high glucose levels, we don't need glycogen phosphorylase to function anymore because we already have enough glucose. We don't need to be breaking down more glycogen. So what are we going to do? We're going to inhibit this enzyme. How do we inhibit this enzyme? Well, the simple way to put it, we utilize a phosphatase, and the phosphatase is going to remove that phosphate group, which is found on the serine, and as a result, it becomes less active or it becomes inactivated. But what we have to understand is that these phosphate groups, they are not accessible to the phosphatase. Essentially, they look more like this. They're not readily accessible to the phosphatase. So what glucose does is that it binds at an allosteric site. Remember, the allosteric site is a site that is different from the active site. And when it binds to the allosteric site of glycogen phosphorylase, it causes a conformational change in the enzyme. This conformational change in the enzyme exposes the serine phosphorylated residues, which makes it easier for the phosphatase to cleave off that phosphate bond and then once again make it less active. So once again, to recap, what does glucose do? It's an allosteric inhibitor of glycogen phosphorylase. It binds to the allosteric site, causes a conformational change which exposes the phosphorylated serine groups so that the phosphate group can be hydrolyzed and we can regain that tense state, which is less active compared to the more active state of glycogen phosphorylase. And that is essentially the regulation. It's pretty intuitive when you think about what's going on. If we have low glucose levels, of course we're going to need it active. But if we have high glucose levels, we're going to want this inactive. Because why? Glycogen phosphorylase is responsible for cleaving glycogen into glucose 1-phosphate monomers so that it can enter glycolysis to produce energy.